This slide presentation reviews vitamin B12. B12, also called cobalamin, was interestingly the last vitamin to be discovered in 1948. There are several cobalamin compounds, each with an atom of cobalt in the center of what is called their corrin ring. Since we have already covered the role of vitamin B12 in 1-carbon metabolism, and specifically methionine synthesis, this video will focus primarily on the food sources of B12 and its unique processes of digestion and absorption. Vitamin B12 is only found in whole foods through animal products, therefore vegans will need to obtain this vitamin through supplementation. The video ends with an outline of the RDA values for this nutrient. We'll first look at food sources of vitamin B12. We see among all of these sources that the food-based sources will be only animal-based products. We see that some foods are fortified with vitamin B12 and there are also vitamin B12 supplements. However, foods naturally containing vitamin B12 will again be from animal products. Therefore, individuals such as vegans who do not eat animal products will not be consuming vitamin B12 naturally found in foods, so they'll need to make sure to consume adequate B12 in fortified foods and or supplements. We'll look at the digestion and absorption of vitamin B12. So the process of digestion and absorption of this nutrient is actually fairly unique and different from many of the other nutrients that we've discussed. So there's a number of factors that actually lead to its digestion and absorption being unique. So we'll talk about the process. So we see that first, once consumed, B12 is often going to be bound to some structure or protein or another compound, so that will need to be hydrolyzed. Certain enzymes or key enzymes involved with hydrolyzing B12 from being bound to different compounds such as proteins would include the enzyme pepsin, but also hydrochloric acid within the stomach. So once the B12 then is hydrolyzed from its bound compounds, then it will be B12 then in the stomach in the free form. So once vitamin B12 is circulating freely in the stomach, it will next bind to a structure called the R protein. So we see in this diagram the binding of free B12 to R protein. In the stomach, we see that B12 is now entering in the free form, and then we see our protein is synthesized in the stomach, and then we see the binding of vitamin B12 with R protein, and then the B12 R protein complex then passes through the stomach into the small intestine. It is thought that B12 binds with R protein to limit its interaction with bacteria or other compounds that may affect it within the stomach. So as it passes through the stomach into the small intestine, pancreatic proteases cleave the bond of the B12 R protein complex, thus freeing both the R protein and the B12. From there, we see B12 that passes through the remaining portion of the small intestine, and during the process, we see the binding of B12 to what is called intrinsic factor. We see here that intrinsic factor, like our protein, is also produced in the stomach. So we can see here the description of what's occurring in the duodenum. So we talked about how that R protein was hydrolyzed from the R protein B12 complex. This frees B12, also known as cobalamin. And then the cobalamin or B12 binds to that intrinsic factor. And the intrinsic factor, again, is a glycoprotein that's synthesized in the gastric parietal cells of the stomach. So again, it's a glycoprotein synthesized by the gastric parietal cells of the stomach. Next, we'll see that the B12 bound to intrinsic factor forms this B12 intrinsic factor complex that continues to pass through the small intestine into the ileum. It is at that point that we have the absorption of B12, but the absorption of B12 is unique, as we mentioned, as compared to other nutrients, in that it is absorbed as a complex bound with intrinsic factor. So what happens is that the B12 intrinsic factor complex binds to an intrinsic factor B12 complex receptor called cubilin. And we can see here in this diagram the movement of the B12 intrinsic factor complex into the ileum, where we have that receptor which binds the B12 intrinsic factor complex. From this point where we see the binding of the complex to the receptor, the B12 is actually absorbed through endocytosis. So this complex is endocytosed into the enterocyte. Once in the enterocyte, the B12 is released from the B12 intrinsic factor complex. So in the enterocyte, we have free B12, and that free B12 is actually bound to another protein called transcobalamin. Transcobalamin, and there's different forms of transcobalamin, but each of the forms are involved in transporting B12 through circulation. So in the enterocyte, B12 binds to transcobalamin. That complex then leaves the enterocyte through the basolateral membrane, and then from there we see circulation of B12 bound to transcobalamin through the blood. Typically, B12 status is affected by the body's ability to absorb B12. And based on the more complex process involving the absorption of B12, which includes the binding to R protein, the cleaving of this complex, then the binding of intrinsic factor, then the binding of the B12 intrinsic factor complex to the receptor. So based on the number of steps involved in this process, there are a number of ways where it can be affected, which may limit the absorption. So there's a number of factors affecting the absorption of B12. These factors include the functioning of the cubilin receptor, also the body's ability to produce intrinsic factor, also the level of stomach acidity can affect this process, as well as other medical conditions affecting B12 digestion and absorption. We'll now talk about the functions and mechanisms of action of B12. So we discussed previously B12's involvement in the process of 1-carbon metabolism, specifically in the synthesis of methionine. So we saw in the folate metabolism, we saw that 5-methyl tetrahydrofolate transfers a methyl group to cobalamin. From there, cobalamin is converted to methylcobalamin, and methylcobalamin transfers its methyl group to homocysteine, and then with the enzyme methionine synthase, homocysteine is converted to methionine. So we see that as a key function and a key role of vitamin B12 in the synthesis of methionine. However, we also see the involvement of vitamin B12 in the conversion of L-methylmalonyl-CoA to succinyl-CoA, and we have this compound, the adenosyl cobalamin, involved in this process. We remember from previously that it was the propional-CoA derived from the oxidation of odd number fatty acids, also from amino acid catabolism. So it was that propional-CoA that was then converted to this methylmalonyl-CoA and then to succinyl-CoA. We'll now discuss the RDA for vitamin B12. So we see that the RDA for vitamin B12 is very low. It's in the units of micrograms, and it's very low in terms of the amount of micrograms that are needed for adults in pregnancy and lactation. So in adults, we see that's 2.4 micrograms per day. In pregnancy, it's 2.6, and lactation, 2.8 micrograms per day. We do have some individuals developing deficiency of vitamin B12. So we mentioned previously it's particularly those individuals that have difficulty absorbing vitamin B12. And there's a number of reasons why someone may have their ability to absorb vitamin B12 be compromised. So the amount of intrinsic factor that is produced, the activity of the receptor used in the process of absorption. Also, the stomach acidity or other medical conditions can limit one's ability to absorb vitamin B12. So that's going to be a primary risk factor of deficiency. So deficiency associated with B12, one main deficiency syndrome is called pernicious anemia. So it's a specific type of anemia related to vitamin B12 deficiency. And pernicious anemia is a type of macrocytic megaloblastic anemia. And so again, we see it's usually caused by inadequate absorption of vitamin B12. However, in the beginning of this slide presentation, we also said that vegans who do not eat animal products are going to have risk of low intake of vitamin B12. So if they don't make an extra effort to consume fortified foods with B12 or supplements with B12, they may also be at increased risk for deficiency. It's important to note that vitamin B12 actually can be stored in the body at fairly high levels. It's stored mainly in the liver, and this is unique for water-soluble vitamins. So typically we see the fat-soluble vitamins being stored. However, vitamin B12 can be stored in various tissues, again, particularly the liver. So it can take months or even years to develop a deficiency. But if someone has a chronic condition or chronically has low intake, they will again be at risk for deficiency, particularly this pernicious anemia.